Snake venom, a cocktail of toxins, has captivated humanity for centuries. This lethal substance, produced by some of the most feared creatures on Earth, has a dual nature that both terrifies and fascinates us. From ancient civilizations to modern science, the allure of snake venom has never waned. This potent concoction, feared for its deadly potential, is also a source of endless fascination for scientists. Researchers dedicate their lives to understanding its complex composition, hoping to unlock secrets that could benefit humanity in unexpected ways. Understanding the science behind snake venom unveils a complex world of biological weaponry and reveals potential medical marvels hidden within its deadly embrace. Each component of venom has a specific function, targeting different systems within the body, making it a sophisticated tool of nature. Snakes, masters of stealth and precision, have evolved a sophisticated arsenal for subduing prey and defending themselves. Their venom is a key part of this arsenal, allowing them to immobilize and digest their prey efficiently, while also serving as a formidable defense mechanism. Venom, a key weapon in their evolutionary armory, is a testament to nature's ingenuity. It is a complex mixture of proteins and enzymes, each playing a role in the snake's survival strategy. This deadly elixir is a result of millions of years of evolution. This intricate blend of toxins, each with a specific target, disrupts the delicate balance of the victim's body, often with lethal consequences. The venom can cause paralysis, internal bleeding, and even death, depending on the species and the amount of venom injected. The fear associated with venomous snakes is deeply ingrained in human consciousness, reflected in mythology and folklore across cultures. Snakes have been both revered and feared, often symbolizing danger, death, and rebirth in various traditions. From ancient tales of serpent deities to modern-day horror films, venomous snakes continue to evoke a primal fear, a testament to their potent venom. This fear is not just a product of imagination, but a response to the real danger these creatures represent. However, beyond the fear lies a fascinating world of scientific inquiry. Researchers are delving into the molecular structure of venom, discovering how its components interact with human biology, and exploring its potential uses in medicine. By unraveling the mysteries of snake venom, scientists are not only gaining a deeper understanding of these fascinating creatures but also unlocking potential avenues for medical advancement. Venom research has already led to breakthroughs in pain management, blood pressure regulation, and even cancer treatment. The study of venom components has led to the development of life-saving antivenoms and opened up exciting possibilities for treating a range of human diseases. As we continue to explore this deadly elixir, we may find that it holds the key to curing some of humanity's most challenging ailments. Snake venom, far from being a single entity, exists in a stunning variety. Each drop of venom is a complex mixture of proteins and enzymes evolved over millions of years. Each species of venomous snake produces a unique venom cocktail tailored to its specific prey and environment. This evolutionary adaptation ensures their survival and efficiency as predators. This diversity, while fascinating, also complicates the study and treatment of snakebites. Researchers must understand the specific venom composition to develop effective antivenoms. Broadly speaking, snake venoms can be categorized into three main types based on their primary modes of action: neurotoxic, cytotoxic, and myotoxic. Each type has distinct mechanisms and effects on the body. These categories, while helpful for understanding the general effects of venom, often overlap, as many venoms exhibit a combination of these toxic properties. This overlap can make treatment more challenging. Neurotoxins, as their name suggests, target the nervous system, disrupting the transmission of nerve impulses. This can lead to severe consequences if not treated promptly. This disruption can lead to a range of debilitating effects, from paralysis and respiratory failure to sensory disturbances and seizures. The speed and severity of these symptoms can vary. The potency and specific effects of neurotoxins vary greatly depending on the snake species and the specific composition of its venom. Some neurotoxins act quickly while others may have delayed effects. Cytotoxins, on the other hand, focus their destructive power on cells. They attack cell membranes, causing cells to rupture and die. These toxins disrupt cell membranes, leading to cell death and tissue damage. The extent of the damage can vary based on the amount of venom injected and the location of the bite. While their effects are often localized to the bite site, they can also have systemic consequences, particularly in severe envenomations. This can lead to complications such as organ failure. Interestingly, the ability of cytotoxins to destroy cells has garnered interest for their potential application in cancer treatment. Scientists are exploring ways to harness these toxins to target and kill cancer cells selectively. Neurotoxins, the most feared components of many snake venoms, exert their deadly effects by hijacking the nervous system. These potent substances have evolved over millions of years, making them highly efficient at incapacitating prey and deterring predators. The complexity and precision of these toxins are a testament to the evolutionary arms race between snakes and their targets. These toxins, often with remarkable precision, target specific components of nerve cells, disrupting the delicate balance of neuronal communication. By interfering with the normal function of these cells, neurotoxins can cause a range of symptoms, from mild tingling to complete paralysis. The result is a cascade of neurological disturbances that can quickly overwhelm the victim's body. This disruption can lead to a loss of coordination, muscle control, and even consciousness, depending on the type and amount of venom injected. One of the primary targets of neurotoxins is the neuromuscular junction, the critical synapse where nerve cells communicate with muscle cells. This junction is essential for initiating muscle contractions, and any interference can have severe consequences. By blocking or disrupting the transmission of nerve impulses at this junction, neurotoxins effectively paralyze the muscles, rendering the victim helpless. This paralysis can be temporary or permanent depending on the nature of the toxin and the effectiveness of medical intervention. This paralysis, if it affects the diaphragm, the muscle responsible for breathing, can be fatal. Without the ability to breathe, the victim can quickly succumb to asphyxiation, making prompt medical treatment crucial. Cobra venom, renowned for its potent neurotoxic effects, contains alpha neurotoxins that bind tightly to nicotinic acetylcholine receptors at the neuromuscular junction. This binding is so strong that it can be difficult to reverse, even with antivenom. This binding prevents acetylcholine, the neurotransmitter responsible for muscle contraction, from binding to its receptors, effectively blocking nerve impulse transmission and leading to paralysis. The victim's muscles become unresponsive, leading to a rapid decline in physical capabilities. Another class of neurotoxins, found in mamba venom, targets a different component of the nervous system: voltage-gated potassium channels. These channels are crucial for resetting the electrical state of nerve cells after they have fired. These channels play a crucial role in regulating the electrical activity of nerve cells. By ensuring that nerve cells can return to their resting state, they help maintain the proper function of the nervous system. By blocking these channels, dendrotoxins, as they are known, disrupt the delicate balance of ions within the nerve cell, leading to hyperexcitability and uncontrolled firing of neurons. This can cause severe neurological symptoms that are difficult to manage. This uncontrolled neuronal activity can manifest as seizures, muscle spasms, and potentially death. The severity of these symptoms depends on the dose of venom and the speed of medical intervention, highlighting the importance of understanding and respecting these powerful natural toxins. Section 4. Cytotoxins, Cellular Destruction and Healing Potential. Cytotoxins, while often overshadowed by their neurotoxic counterparts, possess a unique power, the ability to destroy cells. This destructive capability, while seemingly detrimental, has piqued the interest of researchers exploring their potential applications in medicine, particularly in the fight against cancer. The mechanism of action of cytotoxins involves disrupting the integrity of cell membranes, the delicate barriers that enclose and protect cells. This disruption can occur through various mechanisms, including the formation of pores in the cell membrane, the breakdown of phospholipids, the building blocks of cell membranes, or the activation of enzymes that degrade membrane components. The consequences of cell membrane disruption are dire for the cell. The delicate internal environment of the cell is exposed to the external environment, leading to an influx of ions and water. This influx disrupts the osmotic balance, causing the cell to swell and eventually burst, a process known as lysis. While the cell-killing ability of cytotoxins is undoubtedly a cause for concern in envenomation, it also holds promise for treating diseases characterized by uncontrolled cell growth, such as cancer. Researchers are investigating the potential of using cytotoxins to target and destroy cancer cells selectively, sparing healthy cells. Section 5. Myotoxins, targeting the muscles. Myotoxins, as their name suggests, have a particular affinity for muscle tissue. These toxins, found in various snake venoms, disrupt muscle function, leading to weakness, pain, and in severe cases, muscle necrosis, the death of muscle tissue. The mechanisms by which mycotoxins exert their effects are as diverse as the snakes that produce them. Some mycotoxins, such as crotamine, found in the venom of the South American rattlesnake, Crotalus dorissus terrificus, act directly on muscle fibers, causing irreversible depolarization. This depolarization, a disruption of the electrical charge across the muscle cell membrane, prevents muscle contraction, leading to paralysis. Other mycotoxins, like phospholipids A2, target the cell membrane of muscle cells, disrupting their integrity and leading to cell death. These enzymes, found in various snake venoms, break down phospholipids, the essential building blocks of cell membranes, compromising the structural integrity of muscle cells. The effects of mycotoxins are often localized to the area of the bite, causing pain, swelling, and tissue damage. However, in severe envenomations, mycotoxins can enter the bloodstream and affect muscles throughout the body, leading to systemic effects such as muscle weakness, rhabdomyolysis, a condition involving the breakdown of skeletal muscle, and even kidney failure.